Hi students, today we are going to discuss about one more important short notes from liver function test, LFT, right? Very small topic. Laboratory tests done to differentiate jaundice, right? You know we have three types of jaundice, hemolytic, obstructive and hepatic. How we are actually identifying these three types? We have one test, the name is Van den Berg test. VANDEBERG. Van den Berg BERGH test. Right. In this test, what we are doing is we are adding that Ehrlich diazo reagent or diazotized sulfonilic acid or sulfonilic acid plus sodium nitrate. The last one is very simple. Sulfonilic acid plus sodium nitrite, right? After adding the patient's urine with this sulfonilic acid and sodium nitrite, you will get reddish purple, that is radish purple color or sometimes purple color. This is called azobilirubin. The first formed bilirubin is color giving bilirubin is azobilirubin. Then they will go for analysis by photometry, some 540 nanometer like that. After that, you will get three types of colors. One is color, another one is colorless, another one is initially mild color. After that, intensity will be high. So in the first reaction is direct bilirubin or direct reaction. That is, the conjugated bilirubin react in Vandenberg test is positive. Conjugated bilirubin gives positive Vandenberg test. It is called direct bilirubin. You understand? Direct and indirect bilirubin we are having. Direct means in the Vandenberg test, the conjugated bilirubin gives positive color means direct bilirubin. Whereas for unconjugated bilirubin, Vandenberg is positive means the result is unconjugated bilirubin. This is called indirect bilirubin. Direct means conjugated bilirubin. Indirect means unconjugated bilirubin. You know the meaning of conjugate and unconjugated bilirubin. Before liver, unconjugated bilirubin. After the liver or in the liver, conjugated bilirubin. So if the indirect bilirubin present means what will be the condition? Direct bilirubin present means what will be the condition? That is the logic for this test. Van den Berg test. So sometimes what happens is biphasic test. Here what happens is initially the color is present but mild color. After adding alcohol, the intensity of the color is high. Here, if the color is directly present means direct bilirubin. If the color is after adding the alcohol is present means indirect bilirubin. This is the second point. Direct bilirubin means conjugated bilirubins. Directly it will give color. Indirect bilirubin means there will be no color. You have to add some alcohol to that sample, then only the color will be there means the urine contains unconjugated bilirubin, that is indirect bilirubin. Okay. Third one is biphasic. Initially mild color is there. After addition of the alcohol, the color intensity of the color will be more. Right? So three types of jaundice. The last one is hepatic. So in Vandenberg test, direct bilirubin means, that is conjugated bilirubin means obstructive jaundice, right? Because after liver only the bilirubin will be excreted through the bile duct into the gallbladder. So if the bilirubin is direct means the problem is in the gallbladder obstructive jaundice. Next one is unconjugated bilirubin. The bilirubin is not entered into the liver. Before the liver only you will get unconjugated bilirubin, that is called indirect bilirubin after adding addition of alcohol. This is for hemolytic jaundice. Sometimes what happens is initially mild color, after adding alcohol the intensity will be more means that will be for biphasic jaundice or hepatic jaundice. Okay. Next one is, this is for serum bilirubin. Is there any test to find out in the urine? Yes, we have some test. Urine bilirubin, urobilinogen, bile salts. Three tests we are having for this very important three components: urine bilirubin, urobilinogen, and urine bile salts. Urine bilirubin is tested by Foucher's test or Fouch test, Fouchet, Foucher's test. Urine and fecal urobilinogen is identified by Ehrlich's test, Ehrlichs, Ehrlich's test. This is for urobilinogen and urine urobilinogen and fecal urobilinogen. Then next one is urine bile salts. You already know the name, Hayes test for bile salts. If it is present means obstructive jaundice. The patient clinical scenario is obstructive jaundice. So urine bile salts, what is the test? Urine and fecal urobilinogen, what is the name of the test? Urine bilirubin, what is the name of the test? These are all very important for your liver function test. Next one is liver enzyme assay. Some examiner will ask in your viva, name the liver enzymes. You already know the enzymes. 2 transaminases, 1 phosphatase. 2 transaminases are AST and ALT, that is aspartate transaminase and alanine transaminase. Another one is alkaline phosphatase. This is very important marker for liver injuries because this is elevated always in obstructive jaundice. The next one is 5-nucleotidase. This is also will be elevated in obstructive jaundice. So this is coming under liver enzyme assay. So serum bilirubin you have to mention, urine bilirubin, urine and fecal urobilinogen, urine bile salts, and liver enzyme assay. These are all the very important points for your liver function test. Okay.